Where's Paul?
Track. If you knew the answer, why did you?
Because I realized it after I said it. Okay, so.
I hate it when that happens,
Is
though.
Travis in track too?
Probably.
Track.
Track my back. I was noticing you were closer.
All right, please close your computer lids.
I got to get my moose. My moose.
He's forever sunk. Help! I've fallen and I can't get up.
Sorry.
On the board, we've got, once again, the central dogma of biology, which is DNA's got to do what? Replicate, okay? Because what are its three jobs? To transmit, store, and...
Protect. No.
Copy itself, okay? So that's its job. So what are the two steps? Okay? Replicates by itself, but what are the two steps to do this gene expression? Transcription and translation. Okay? So those are the two steps to get there. And now we bring in a second nucleotide besides nucleic acid besides DNA. What expresses the genes, the DNA? Well, there's another molecule called RNA, but ultimately the gene is expressed as a what? Protein. Okay? The gene is going to be expressed as a protein. And so, how many bases code for one amino acid that makes up our protein?
3.
3 bases code for one amino acids. Okay? How many amino acids are there, though? 20. There's 20 different ones. Okay? So we have to have that. And what do we call that if it's a DNA-based sequence? And the 3 bases that code for a protein, I just said it. We call that the code. Okay?
Essential code.
But what do we call the complementary base pairs that are made from that for messenger RNA? What do we call this?
The
codon.
Yeah, the codon.
And then what is on the bottom of each of the transfer RNAs that brings the correct amino acid during translation?
The
The
nucleus.
anti-codon. So we got the codon, and anti-codon.
Oh.
And they're all 3 bases, okay? And they all mean one amino acid. When you use that table like we were using yesterday, which base triplet do you look up? The codon, or the anti-codon? It was a chart of codons, right? It was a chart of codons. All right. So, hey, if what's mutation?
Mutation is the genetic something goes wrong.
Yeah, something's wrong with
Genetic code.
the genetic code. Well, what would that look like?
One or some less proteins.
Well, not proteins. The genetic code is what? Our DNA. Could I maybe have, during replication, instead of having an A here, I mispaired it and I have a G? And then if it went to make the protein, if I change this A to a G, would that mess up the sequence of messenger RNA?
Yeah.
Would that potentially mess up the sequence of amino acids?
Yeah.
And then would it fold up to the right shape? Maybe, maybe not. Ha! Okay, get over there. You gotta quit that looking off in space, okay? Now, okay? I just realized I'm twins with you and you. Nice. All right, now.
I just realized I'm ginger.
No, not yet. All right. Are you ready? What am I talking about? Mutations. Are all mutations bad, harmful?
Yes.
No, they're not.
Unless you're super.
Yes, Mr. Schulenberg?
I keep forgetting to tell you that LabQuest Mini that does not work.
Yeah?
I emailed them and they should send it in and replace it with one
Which LabQuest Mini that doesn't work?
that you don't have.
I think I got another one that doesn't work.
Uh-oh.
Well, it only works with some computers.
We'll talk later.
We'll talk.
Hi, Mr. Schulenberg.
What are we doing here?
We're talking about the.
Study of life.
We have no idea what we're talking about.
We're talking about Central Code.
Amino acids and
Amino acids.
Yeah, we're talking about like
You know,
the
basic stuff.
No, they're acids. They're not bases. They're acids. There's a big difference.
We're talking about like the Central Code and stuff.
Good one, Mr. Schulenberg. Okay,
I do have a question.
thanks, Mr. Schulenberg. You just totally wrecked my class. All right, so now, everybody here. Okay, so let's think about this. So, in fact, some mutations make no difference at all in the protein because remember how many different combinations of triplets were there from that chart yesterday? Did anybody, you might not have counted them up, but I'll tell you, there was a total of 64 different combinations of base triplets. But how many amino acids are there? There's only 20. Do you remember which amino acid had 6 codons for it? The pirate? Arg. Arg. It had 6 codons for it. So maybe if I substitute one of those letters, it won't make a difference ultimately in the amino acid. Okay, so sometimes I'll get a different amino acid and it won't make a difference. Sometimes is it beneficial? Yeah. Sometimes are they harmful? Yeah, so they can be harmful. They can be silent mutations, meaning they don't show up. Okay? So now watch what would happen if what if I totally left this one out? Boom.
Oh my gosh.
Okay, which would be more harmful, if I completely deleted one of these, or if I just substituted them on one?
Completely deleted.
Because what would it do to my base codons? It would shift them all over, right?
Yeah.
And that's called a frame shift mutation. And so would that affect all the amino acids after that point?
Yeah.
Yeah, and so that the chance of that being more harmful is going to be higher because we would shift that frame out. More on mutations on Monday. Here's what we're going to do today. You know that test you took on the last test we took?
Mm-hmm.
Okay, I'm going to give you a chance to make corrections on that today. Okay, so
Does that mean we did bad?
did we do bad?
Yeah.
No, actually we did better than the test before that.
Sweet.
So hit that one, and it's not in the grade book yet. So we'll make test corrections. I'm also going to send you to a website, and can I have your attention, please?
No.
Do you guys call them fireflies or lightning bugs?
Both.
Does anybody call them fireflies? Is that just like here in Indiana,
I call them
they're
fireflies.
lightning bugs? But you call them fireflies.
So does
Nice.
Cole.
Cool. Okay. Are they the same thing? Yeah. Okay, now here's the question for you. Today, you're going to learn why a lightning bug
Glows.
lights. What does he have in his rear end that he can glow his butt and you can't?
We don't.
Okay, so
I wish we could do that with our
we're going to
butts.
find out today why that happens. Shh, shh, shh. Okay, I'm going to give you a hint. They make a protein that glows. Oh.
Can we do that?
Oh. Okay. They make a protein that glows. Well, if they make a protein, what do they have that tells them to make the protein? DNA. They have a gene for it, right? Okay. So we're going to do that. First off, though, we're going to watch a couple things.
You.
Is the stuff in a lightning bug the same thing as like in a jellyfish or an
No.
anglerfish?
It's different
No, wait a minute. How comes they can do it but we can't?
because they make the right proteins and we don't.
Oh. Rats. That would be awesome if we could do that.
Make a cow.
Your screen, your projector's off the screen a little bit.
Four different embryos and cows.
Tutatube.
Oh.
I love how fast the video loads for you.
I know.
Okay.
My grandpa told me about experiments they used to do where they replace the cells.
Some of you had trouble with your book again, and you might not have gotten this tutor tube loaded, so close your laptop. Let's watch this together because what she has to say is very important. So stay awake and go.
Remember what Mendel said? There's
No.
what you see, then there's what you get, genetically speaking. Well, maybe he didn't say that, but it's true. What you see as physical traits is your phenotype. What you get from your parents is a mix of genes. That's your genotype. An organism's genotype, or genetic makeup, is determined by its DNA sequence. But how exactly does the genotype determine an organism's phenotype? It's all in the proteins. Biologists often call this the central dogma. DNA makes RNA, makes protein. So why is this process central to molecular biology? It's because this process explains how proteins are synthesized, and proteins determine phenotype. They're essential to every aspect of cell life. About 15% of the mass of your body is proteins. Proteins are the main components of your muscles, cartilage, ligaments, skin, and hair. Feathers, hooves, and shells are mostly protein too, but you don't have those. Plants rely on proteins too. Important molecules such as insulin, antibodies, and hemoglobin are proteins. In addition, enzymes are proteins. Enzymes catalyze or speed up and regulate chemical reactions such as DNA replication and transcription, and most of the reactions in cell respiration and photosynthesis. An enzyme can cause a reaction that produces colored pigments in flowers and controls tissue differentiation, and so much more. If a plant contains an allele that encodes a protein, that creates a red pigment in flowers. Then the flower's phenotype will be red. This fact will be important to know on prom night. Proteins are important. So the central dogma is so central because the function of DNA is to code for the instructions to build proteins. And proteins determine phenotype and are necessary for almost every aspect of cell life. And all living things are made of cells.
All right, that was just an awesome review of all of biology. And you need to review that for the end of course
Assessment.
assessment coming up, okay, which will be your final exam. Now, how many of you got the interactives to work?
Yes.
Okay, if you didn't do those interactives, you do need to go back and do that today. So let's just take a look at this to review this whole process of making a protein.
That was fun.
The whole process of making a protein starts where? In the... No, we're not going to have replication of DNA when we want to make a protein. What are we going to do? We're going to make RNA. Okay, so, good point, Stephen. Is DNA replication and gene expression two separate processes? Yeah, okay. They're similar but different. That made sense. What's the first part of this process of ultimately make a protein? We're going to start with the DNA, right?
There's the DNA.
There's the DNA, okay? And there's what's our smart molecule adding basis? Polymerase, okay? And that would be called what? What did we just watch? Transcription. That messenger RNA that was just made, we're watching it again. Where does it go? Where does it go? Where does it go? Out the nucleus to the ribosome. The cytoplasm, where you find the ribosomes. Here's your, so far, how many types of RNA have we seen?
2.
Messenger RNA and? No, not transcription RNA. Transcription is not RNA.
Transfer.
We have not seen transfer RNA yet.
Messenger RNA.
That's the messenger RNA. Here is the mRNA. Here's the next one. The second one is what? The actual ribosome. That's ribosomal RNA. And then we have translation. Where's this one going?
He doesn't know.
Oh, yeah, he knows.
Go for him.
Is it going to go get another amino acid?
Mm-hmm.
Yeah, so what are these things? Transfer amino acids. We're on the inside of the ribosome. The ribosome is just a place so it holds the transfer RNAs together so that we can start hooking our amino acids Okay, if you didn't have the ribosome, you wouldn't have a place for this to happen, to hold them together, so you would never get any proteins. So, oh, what are these 3 bases down here at the bottom?
Codon.
Oh, no, those aren't the codons.
Those are the
Anticodons. They are the opposite of the codon, which is down here in our messenger RNA. Okay, so each transfer RNA will only get one type of amino acid, and the type of amino acid it gets is based on its anticodon. So we're building our protein, and eventually, what do we call your new little protein that you make? What?
polypeptide.
A polypeptide. Okay, so once we get our protein, there's our polypeptide. All right, so did you do this little interactive?
Yes.
Where you practice base pairing?
Mm-hmm.
If you didn't get it to work, that's what you're going to do today. Okay?
Why are you making this?
Remember, if you're making RNA, do you use T or U? You substitute U for T. Okay, and then we're out here at our ribosome, and now you're physically dragging the. How do we know if this works or not?
You have to look for the right shit.
They have to be the right shapes, don't they?
Yeah.
Oh, and the right shapes are complementary. That's how your body doesn't really look and say, oh, there's an A. It's really by the shapes. Okay, the shapes are complementary, and that's how it recognizes each other. It's all by the shapes of the molecules. So you could have done that. Now I want to answer these questions right now.
Why does it use a U instead of a T?
God made it that way. Where does transcription occur?
And the nucleus.
Nucleus.
Nucleus or cytoplasm? Where does transcription occur? Nucleus. Transcription happens in the nucleus. Where does translation occur? At the ribosome, which is in the cytoplasm. Second one, what role does DNA play in the transcription process?
It plays the messenger.
It's the template.
No, it doesn't do the reading. It's the, and you got to use this word. It's the template. What's that mean? It's what gets read, okay? Kian, leave us here alone. Okay, so now the third question. What's the difference between mRNA and tRNA? Which one's carrying an amino acid? mRNA or tRNA? tRNA. So what's the T stand for in the tRNA? So what are we going to transfer? An amino acid. Okay. Which one is kind of like a little blob, kind of like a little key thing? The transfer, which is just a single ribbon. Messenger RNA. What is messenger RNA? It's the copy of the gene, or the transcript of the DNA. What role do ribosomes play in translation? I just told you this. Why do you have to have ribosomes? Close this. We're answering.